Okay, now still dealing with probability, Punnett squares, and Mendel's principles. Today, as I mentioned in the video that preceded this, we are going to be dealing with something a little more complex, and that we are going to be dealing with two-factor traits. In other words, we're going to deal with two traits at the same time. So you can see in this one, we are looking at, in this particular Punnett square, the color of the P and the shape of the P. Okay, so in that case, we're going to deal with genotypes consisting of 4 letters, 2 for each trait. In our previous Punnett squares, we only dealt with a genotype of 2 letters. Today, again, I want to emphasize 4 letters for each genotype. Now, something you need to know, and something that comes into play here, and this is going back to Mendel, is the law of independent assortment. And what does that law of independent assortment tell us? It tells us that one trait has no bearing on the outcome of another trait. In other words, genes for different traits can segregate or separate independently during gamete formation. And again, this gamete formation occurs during meiosis. So again, the best way to do this, I believe, is to simply work some examples with you and go from there. So, Punnett squares for two-factor crosses. Okay, for two-factor crosses. Again, first step, we're going to use the same steps we used in the previous lesson. In step 1, we have to identify the dominant and recessive alleles. Now we have to do this for both traits. Okay, so what we are going to do is look at flower color and stem length. So we have to have a letter for flower color. In this case, I chose P because P, purple. Purple is the dominant trait in flower color here, especially when dealing with pea plants. And I chose the letter T because tall is the dominant trait in pea plants over short. So again, we're now, instead of dealing with 2 letters, capital and dominant, we're going to deal with 4 letters. 2 capital letters, 2 lowercase letters. That is step 1. Step 2, write the genotypes of the parents. Again, the genotypes are going to consist of 4 letters. 4 letters. In this case, both our plants are heterozygous. So you have to ask yourself again, what does heterozygous mean? Okay, heterozygous means that the alleles for that particular trait are different. In other words, you need 1 capital letter and 1 lowercase letter for each of the traits. So both of our plants are going to have a big P and a small p for flower color. And they're both going to have a big T and a little t for their stem length, because again, they are heterozygous for both traits. That is step 2. Step 3, that is making the Punnett square, or setting up the Punnett square. Determine what alleles will be found in all the possible gametes. So, those are all the possibilities for plant 1's gametes. We can have a big P go with a big T. We can have a big P go with a little t, a little p with a big T, or a little p and a little t. Again, this is Mendel's law of independent assortment. We go to plant 2. Plant 2 has the same genotype, so it's going to have the same possible gametes. From here, again, emphasize law of independent assortment. Again, we go and we do steps 4 and step 5. Step 4 here, we're going to draw a table with enough spaces for each pair of gametes and from each parent. And then we're going to enter those alleles of the gametes to produce both parents at the top and left of the thing. Okay, now here is a big difference, or a second big difference. In this case, we're going to need a Punnett square consisting of 16 boxes. Again, a Punnett square consisting of 16 boxes. Now, we take what we got from step 3, and we're going to take the first plant and place those gene pairs across the top. So there's our big P, big T, big P, little t, big little p, big T, and little p, little t. That is the plant 1. Plant 2 goes down the left-hand side. We do the same thing here. Again, this is the part that many of you are leaving out when you are working your Punnett squares for the 1-factor crosses. It is important to put these alleles or gene combinations around the Punnett square. Now, just like we did before, we're going to join alleles together. In other words, we're going to put the P's back together, and we're going to put the T's back together, and we're going to create possible genotypes for the offspring. So if we look at go to our next part here, this is step 5. We're going to combine the alleles into the Punnett square. So if we look at this first one, the 2 big P's come together, the 2 big T's come together. Again, we're putting P's with P's, T's with T's. Very important to do so. It makes it easier at the end to do step 6. We do the P's together, so it gives us 2 capital P's. We put the T's together, we get a capital T and a little t. We continue to do this throughout our Punnett square. Do this for each column, each row. And you can see here how the P's are going together and the T's going together, with this very last box getting nothing but lowercase letters. 2 little P's, 2 little T's. Now we move on to step 6. What is step 6? This is calculating probability. Now, for this video, this PowerPoint, I'm going to do genotypes and phenotypes. For your worksheet, I'm actually just going to have you figure out the probability of the phenotypes. Okay, make things a little easier when it comes to that. So, if we go through and we look at the genotypes, there are a variety of different genotypes that came up. There were 9 different genotypes. What you simply have to do is go through your Punnett square and count how many of each one's come. So for the one that was homozygous dominant for both traits, you had 1. Okay, the one that had the 2 big P's and a big T, little t, if you count through your Punnett square, there were 2. You continue doing this, you'll find that there are 2 that have a big P, little p, and 2 big T's. There are 4 that are heterozygous for both traits. We have 1 that has a big P, little t, little t. We have 2 that are big P, 2 little t's. 1 with 2 little p's, 2 big T's, 2 little p's, big T, little t, and only 1 here that have the homozygous recessive genotype. That takes us to our phenotypes. In each case, and for the problems that you're going to do for me today on your worksheet, there will always be 4 possible phenotypes. In this particular case, we were dealing with flower color and stem length. Purple being the dominant flower color, tall being the dominant stem length. So when you look for anything, and remember we're dealing with Mendel here, so we're dealing with complete dominance. Okay, anytime you see a big P, you're going to have a purple flower. Anytime you see a big T, you're going to have a tall stem or a tall plant. So you're going to go through and see how many of the 16 boxes have a big P and a big T in them. If you take the time to do that, you will notice that 9 of those genotypes have a big P and a big T. Right here is 1, there's 2 more, there's 2 more there, 4 more here. Okay, that is a total of 9. So we've just taken care of these 9 individuals. Now, in the next part, we're looking at purple flower short plant. So that is a big P to make it purple, but in order to be short, you have to have 2 little T's. So we're looking for a big P with 2 little T's. And I look at my genotype numbers and I see 1 and I see 2 more for a total of 3. Now, white flowers. How do I get a white flower? Since white is the recessive color, you can only have little p's. To be tall again, you have a big T. So I do the same thing, 2 little p's with at least 1 big T, and I have 3 of those. Now, as I move on, white flower short plants, those are the 2 recessive traits. In other words, I have nothing but lowercase letters. I look over here and I have 1. So now all you have to do is change that into percentages for the possibility or probability of the outcome. Again, you're doing this out of 16 since there were 16 boxes in your Punnett square. We take 9, divide by 16, and there is a 56% chance that these 2 plants would produce plants with purple flowers and that have tall stems. There is a 19% chance that they'll have purple flowers with short stems. 19% chance they'll have white flowers with tall stems. And finally, there is a 6% chance of doing white or offspring with white flowers and short plants. So again, just a little bit more complicated, but if you take your time, you look things over, very, very doable. Again, remember all genotypes have 4 letters. Make sure you're putting your letters together of the same kind, and you will always have 4 phenotypes. Make sure you watch the next video before moving on to your assignment.